Hello everyone, this is Stacy Elliott with RioSense. Thank you for joining our webinar today. As you know from the advertisement, what we're going to talk about today is how you can use viscosity measurements to guide you as you're developing your formulation. And although it's not evident in the title, I think it was evident from the advertisement information that we're really going to focus on proteins today. So the data we're going to share is from Model Protein Solutions. So just to quickly overview, the goal of the webinar. What I really like to do when I think about viscosity measurements is how, and I'm going to refer to my flow chart at the top here, is how you can interpret viscosity in terms of the component interactions and the microstructure that exists. So as you develop your formulation and you're adjusting the components, specifically for proteins, if you're adjusting pH, ionic strength, introducing excipients, even adjusting the concentration of the antibody or protein. All of these formulation additives are going to impact what's happening on the molecular level. And by that I mean the nature of the individual molecules, and then also how these molecules are going to interact on the pair level, often thought of as the protein-protein interactions. And then because you can have attractions or associations, then you can generate complex microstructures such as aggregates or even network formation. And so then this all will influence your viscosity measurement. So I kind of want to introduce the idea of thinking in this way, that as you measure viscosity and you know what your formulation components are, there are some ways of trying to interpret and pull out how everything's interacting and the microstructures that are forming. And the ultimate goal, I think, then is to sort of develop a database of information so that as you systematically change something in the formulation, you can see how the viscosity changes and interpret that as a systematic change in the interaction and microstructure. So then you're developing basically you can then control the system as opposed to just doing a brute force, let's just change everything and hope to get what we want. So hopefully that was clear. So as we go through this and think about this type of process, just quickly I wanted to review the basics. I always like to bring up what is viscosity. I know a lot of you do that measurement, but maybe some of you don't. But I always like to highlight what it is so that it's very clear why viscosity is so sensitive to what's happening on the molecular level. And then I want to introduce the idea of colloidal systems and how they're viewed, and how we can start adopting ideas from these systems in interpreting viscosity data. So I know this is actually slightly controversial when it comes to applying the colloidal view to proteins. I've seen a whole variety of comments in the literature, from it's total rubbish to no, my data and my analysis clearly illustrate that this colloidal model works for my protein system. So just ahead of time, I'd like to state that my stance on this is I'm kind of in the middle, I guess. I see the value, certainly from a qualitative point of view and maybe even semi-quantitative, but I do acknowledge that it's not perfect and ideal because of the nature of the proteins. I recognize that there's non-uniform charge distribution. They're not necessarily spherical, or they can have anisotropies. So I do see the differences, but I certainly think in terms of interpreting data and looking for trends that they can have value in many systems. But I am aware of limitations here. So reviewing that, and then we're going to go into the data that we've collected, and then looking at how we've started to analyze the data with respect to how we've adjusted the formulation, and then what this might mean to the interactions that exist, and then how we can interpret the viscosity data from those. So we're looking at our model proteins that we're looking at are the bovine serum albumin, the BSA. where we're going to adjust the pH to control the interaction. So we can adjust pH to control the level of charge, effective charge of the molecule. And then we're going to, once we do this with the different buffers, we're going to monitor viscosity as a function of temperature to look at either whether you refer to it as melting or denaturation of the protein. And then we're going to look at some bovine gamma globulin samples, where in this case we're going to adjust the salt type that we add to the buffer, and then also the ionic strength of each salt to adjust the interactions. And we're going to, in this case, we're going to analyze in a little more detail viscosity versus shear rate and see what type of information we can get back from there. Okay, so just quickly then reviewing viscosity, we know what it is. So viscosity is, it's a defined property that's intended to characterize the resistance to flow, and it is defined as the shear stress divided by shear rate. So what do these things mean? So if you look at a very simple example of a fluid between two plates, where our lower plate is stationary and our upper plate is being pulled along at some particular velocity. From this, we can calculate the shear rate, which is characterizing the extent of the deformation that we're applying. And this is just the velocity gradient that's produced from this type of flow field that is perpendicular to the direction of flow. And so in this very simple geometry, the shear rate is just the speed of the upper plate divided by the height of the fluid. So again, we're introducing deformation. We're going to characterize that deformation by the shear rate. And the shear rate is essentially just a velocity gradient in the fluid, which is perpendicular to the flow field. Now, once we reach steady state, as we're moving this upper plate along, to move the plate, there's going to be some force per unit area required. The per unit area, by that I mean the fluid, the area that's contacted by the fluid. At steady state, this will balance the viscous stress. So that's what I mean by shear stress, is the force per unit area required to drag the plate, which is balancing the viscous stress. So viscosity then is defined as stress divided by shear rate, and this is perhaps a more common form for people where the viscous stress is equal to the viscosity times the shear rate. And then pointing out that the viscosity is going to be nearly always dependent on temperature, and then often, when non-Newtonian, dependent on shear rate. So when you think about what is the steady shear viscosity and why is it so sensitive to what's going on the microscopic level? So as I mentioned, when you set up a shear flow, you're introducing the velocity gradient perpendicular to the flow. So what this means is that each neighboring fluid element is moving at a different velocity. And so you're basically forcing them to slide past each other. And so when people try to speak in general terms about what viscosity signifies, they'll often refer to it as being an internal or molecular friction. And that kind of makes sense if you think about these molecules being forced to slide past each other, particularly when you're introducing higher molecular weight or macromolecules such as proteins or synthetic polymers, or even particles such as colloids. So what we're really going to emphasize today is looking at viscosity and how we can think about the viscosity in terms of what's happening on the molecular or microscopic level. Specifically in terms of how the components are interacting and the types of microstructures that may be formed because of these interactions. Okay, so I then wanted to introduce, when you're dealing with colloidal systems, how do we view these systems, and why is it potentially useful to view protein systems as colloidal systems? So a colloid is generally defined as a particle within a particular size range that can be dispersed in a fluid phase. So the size range is going to be somewhere from 1 nanometer to 1 micron. And this size range is defined such that on the low end, these particles, and for our discussion, we're going to make the analogy between the protein and the colloidal particle. So the protein molecule would need to be significantly larger than solvent molecules so that you could view the solvent as a continuum. But they would have to be small enough so there's an upper bound on the sizing so that they still undergo Brownian motion due to thermal fluctuations in the solvent. So basically they're constantly diffusing around in the fluid. Now, the reason it needs to be, one of the reasons that we need this lower end bound such that the fluid can be treated as a continuum is we want to treat the system. Colloids are generally treated as what we refer to as pseudo one component systems. So we basically have the colloid or the protein or whatever we're assuming the molecule is. We have this, and then we account for everything else that's introduced into the solvent or the buffer by in the description of how these components interact. So we have the colloid or the protein, and then everything else we add just comes in by the description of the interaction potential. So the interaction potential, which I've sort of drawn over here, is sort of the basis for interpreting these types of systems. And so if you look at two of these spherical objects, protein or colloid, we're going to look at how they interact as a function of their center-to-center separation, which we're defining as R. And we're defining the radius of this object, effectively spherical object, as A. And so I've drawn sort of a common form for this interaction potential as a function of separation, although it could be purely positive, which would be purely repulsive, or it could be purely negative, which would be purely attractive. And then it could have other forms as well, where you not only have this primary minimum, but secondary minimums as well. But the way to view this is that positive potentials are repulsions, meaning that as you bring the components together, there's a resistance to that, acting to push them apart. And so you can think of the force is actually defined as the negative of the gradient in the potential. So you can imagine as you're coming in along this potential, if you look at this expression for the force, you can see that there's a repulsive force acting to push them apart. Then when you come over the barrier, now the force is attractive as you move into the minimum of the potential. So this is just a way of characterizing whether you have the repulsions or the attractions and quantifying it. So when you think of a system that has repulsions, then this is something that has a stabilizing barrier. And it tends to make systems what we refer to as kinetically stable. So the time frame for stability is going to depend on the magnitude of this barrier. So higher the barrier, the longer the stability. You don't get something that's thermodynamically stable until this barrier goes towards infinity. But if it's high enough, then, for all intents and purposes, the time frame for stability can be very long. So in this case, you generally have individually spaced particles or proteins that don't form aggregates because there's this barrier to the attraction. However, if your barrier is minimal and you are able to actually enter into this attractive well, then you do start to form aggregates to some extent. And the exact structure that you'll get will depend on the strength of the attraction and the magnitude of the repulsive barrier. So again, the higher the barrier, the more stable that your system is, and less likely you are to form the aggregates, even if the potential minimum does exist. So with this in mind, when you're dealing with when people are thinking about colloidal systems, they tend to want to correlate, all macroscopic behavior, including viscosity, to how the particles or proteins interact, so to this pair potential. And then the belief is that you can control the behavior by making adjustments to this potential. And you can do this by adjusting, in the case of the proteins, adjusting the buffer. So if you accept that the protein or antibody is fixed, and now you want to manipulate things, you're going to need to go in and adjust the buffer. So one of the relevant ideas that we're going to talk about in a moment when it comes to the BSA is a charge system. So one way to get a stabilizing repulsion is to have surface charge on the molecules or particles such that the barrier is sufficient to prevent the aggregation. And so you can generally adjust this barrier by adjusting the buffer conditions, either pH or salt content. So that's kind of going to be the relevant interaction that we're going to deal with today, is an electrostatic interaction. And just a quick background on why a charged species would give you a repulsion. That would be, and so let's talk initially when you're dealing with something where you have a very low ionic strength or minimal added counter ions. So there's some surface charge density on the molecule or particle, and because the system has to be electrically neutral, there's going to be counter ions surrounding that object. And these counter ions, depending on the charge density, will occupy a particular space, distance away from the molecule, giving you a different range on the repulsion. And then the density of them will then give you also a magnitude. And then as you add salt into the system, then you start to basically shrink this layer down, and what's happening is you're generally screening the electrostatics is generally the term that you use for this. So that's going to be the relevant type of interaction for our systems today. Okay, so let's start with the bovine serum albumin and look at, this is quite a well-studied protein, and there's quite a lot of information out there on its behavior, specifically as a function of pH. So this is not my data, this is data that I was able to find in the literature, and I basically just reproduced the plots, not worrying about getting exact values, but I wanted to show you the trends of what is known for this particular protein. So what we know for BSA as a function of pH, we know that the effective, we know the behavior of the effective charge or the charge density as a function of pH. So we know that the isoelectric point, or point where the net effective charge is 0, is close to 5. And then so that means that as we go in pH above 5, we become more anionic, and then as we go below, we become more cationic. So what we'd expect then is that as we deviate from the isoelectric point, either increasing pH or decreasing pH from 5, that we would introduce a larger electrostatic repulsion. So that, let me jump back, this barrier in the potential would be adjusted, and it would increase as we move away from the isoelectric point, so long as we're not adding a lot of extra counter ions by having a high ionic strength. And this idea is also consistent when you look at some other experimental data that's in the literature for the diffusion interaction parameter, K sub D. So just to quickly review, in case you're not familiar with this, the interaction parameter is, if you do so you would generally get this from measuring diffusion from light scattering techniques. And since you'd be looking in the low concentration regime, you would analyze the data by doing a virial expansion of the diffusion coefficient in terms of concentration of your main component, your protein or whatever you're dealing with. And so this diffusion interaction parameter is the first order term in this expansion. And again, you get this by fitting light scattering data, diffusion coefficients that you would get from light scattering data. And I'll also point out that this parameter is defined for what's referred to as collective or mutual diffusion, because there are actually different types of diffusion that exist. Another option is the self-diffusion, but this is for collective diffusion. And so the general understanding is that when this parameter is positive, the main interaction that exists, the dominant interaction, is repulsive. And when this parameter goes negative, the dominant interactions are attractive. So what we can see is that it's attractive or negative in the vicinity of the isoelectric point. And then as we deviate from this pH in either direction, then we become ultimately repulsive, and then more and more so. So that's consistent with the idea of viewing the system as having the interaction potential, which we can adjust by adjusting pH. So let's see. So this is what we know about BSA and how it behaves as we adjust the pH of the system. So now what we want to do is, what can we do with viscosity and how can we correlate it to this information? So in the past, we've shared some data where we've tried to come up with an alternative way of looking at whether we refer to it as the melting temperature or denaturation of the protein. So the point where the protein As we heat it, ultimately the protein will unfold, and then as it unfolds and it can expose different types of functional groups, it can then have a higher likelihood of aggregating. So we've in the past showed that viscosity is a sensitive indicator of this particular transition, and sort of could be an alternative to something such as circular dichroism or differential scanning calorimetry. So we've showed some data in the past for a single buffer composition where we were able to see this transition. So now what we wanted to see is, can we detect changes in this behavior in this transition region as we adjust the buffer, and specifically adjust the pH of the buffer, where we know that we're, based on the literature data, where we're adjusting the interaction potential and the strength of the potential. So what we're doing is we're looking at buffers, we're looking at dilute concentrations of the BSA, so we don't actually gel up in the system. We still have a fluid, but the viscosity can change. And we're going to adjust the pH from something close to the isoelectric point, something that's going to be very weakly anionic, and then systematically move up to higher and higher pHs where we increase the negative charge of the molecules. And what we did then was we monitored the viscosity during the heating cycle, and then we cooled it back down. So we wanted to heat it up to look for the transition and then cool it down so we could see how does the sample recover, what state does it return to? Does it return to its original state, or is it permanently altered due to this process? So what we see then, so let's first look at this top plot where we have viscosity versus temperature, and this is our entire heating curve, where we have, just as a reference point, how the buffer behaves under the same heating process. And of course the buffer is just the simple fluid, and it's a reversible Arrhenius temperature dependence. However, we see that for our BSA and our variety of buffer solutions where we have adjusted pH, we can see that there is a transition slightly beyond 60 C, where the viscosity starts to deviate from the buffer value, and it deviates in a different manner depending on the buffer composition. So this plot down below here is just basically zooming in on this transition region so we can get a better view of what's happening as we for our different buffer values. So again, the buffer alone is just consistently decreasing, which we would expect. And now what we see is that if we start here with the pH 5, so this is very close to the non-charged state or the isoelectric point, and we increase the pH, or essentially increase the charge and then increase this repulsive stabilizing barrier, we can see that the variation that occurs, the magnitude of viscosity change, decreases as we increase pH. So this is consistent with the idea of, if we have a system that's charged and more increasing charge as we increase pH, that as these protein molecules unfold, if there is some stabilizing force, this potential maximum due to electrostatics, then we could expect that there would be less aggregation occurring. So let's take another view of this so we can get a more complete view, I guess. So I mentioned we did the heating and the cooling cycles. So now, just to make it a little simpler, I've just put two of the systems here. So we have pH 6.3 and 7.2. So again, as we go up in pH, we're increasing the charge, increasing the level of stabilization that could occur. So what we see is now we have solid points are heating, and then open points are the corresponding cooling curves. And so in both cases, what we notice is the system does not return, the sample does not return to its original state, but the final state is quite different depending on the pH value. And another way to look at it where the transition is a little more clear is if we look at the relative viscosity as a function of temperature. So relative viscosity is just the viscosity of the protein solution at each temperature divided by the viscosity of the buffer solution at each temperature. And what we see initially is things are very flat, so it's trending with the buffer behavior, and then at some point we deviate and we start to see an increase. And the extent of the increase in viscosity is dependent on the pH, and then also the final state that it achieves when we cool it back down is dependent on the pH. And so again, I wanted to emphasize that what we're seeing here is that when we have a higher pH, where we have a higher charge density of the molecule, and then effectively this would imply that we have a higher potential barrier that could act as a stabilization mechanism. We see less change as it goes through this melting or denaturation. So this is an example of where we can relate our buffer composition to what we expect to be happening on the molecular level, specifically in this case in terms of the pair interactions and how an electrostatic barrier the magnitude is increased or eliminated entirely near the isoelectric point, and then how we can see that manifest in the viscosity data. And so of course you could then do some additional experiments here where you add other excipients to the buffer that are known or expected to control the interaction potential. So one thing that is expected to stabilize proteins are various types of sugars or individual amino acids. So you could also look and see how this transition changes with other components. We just wanted to initially do this experiment with adjusting something that was sort of well documented in the literature, and that would be the pH and how it relates to charge density and then the electrostatic repulsion magnitude. Okay, so excuse me. Moving on then to our next system. So we're also dealing with another commonly investigated model protein, and in this case we're looking at bovine gamma globulin, although now we're looking at the behavior in the concentrated state. So as I mentioned, when we were doing the melting studies, we were looking at dilute protein solutions down around 30 mg per mil. Now we want to look at higher concentration protein solutions. In this case, we're looking at 250 milligrams per milliliter. And one of the reasons we're looking at something much higher here is because ultimately, as you'll see, what we want to look at is the shear rate dependence and what we can learn from the shear rate dependence. And to get samples that shear thin, particularly with the gamma globulin, we need to go to higher concentrations. So in this case, our first group of experiments with this molecule was where we were keeping a single pH and adjusting both the salt type and then the concentration of that salt. And in this case, our pH was slightly above what is known to be the isoelectric point. So that's going to be somewhere in the vicinity of 7, and then we're slightly higher. So this would be very minimal charge, maybe slightly anionic. And so what we have in the plot here is viscosity as a function of ionic strength. And then we've added sodium chloride, sodium bromide, sodium sulfate, and potassium chloride to see how adjusting the salt concentration for these different salts would change the viscosity behavior. So the general trend that we see for all of the salts, although they differ quite a bit depending on the salt type, is the initial decrease in viscosity and then an increase. And the extent of this then seems to depend quite heavily on the type of salt. And although this is a little less well defined than the way I described the BSA, because for these types of protein molecules, when you start introducing the salts, the behavior can be quite complex, and the trends that you see can tend to vary a lot depending on where you are relative to the isoelectric point, and then also even just for the individual molecule in general. So I'm going to speak in this case in more general terms, not so specific as I did for the BSA, because it's a little more complicated. But the thought process behind it is that the reason the different salts behave differently is that you can relate them to what's referred to as the Hofmeister series. And although I only included in my little diagram here the salts that I was using, although the analysis has been done for a whole variety more than I've just explained. But I just wanted to put the ones I had used on here so that we could sort of look at the ranking of the series and then correlate it somewhat to the behavior that we saw. So the general idea is that when you're near the isoelectric point for a number of proteins or antibodies, that initially adding salt will increase the solubility. And I'll point out here that for these particular samples that we prepared, the buffer alone, which had a very low ionic strength due to the phosphate salts, we weren't able to get the gamma globulin into solution. We had to add some low level of our additional salt here to get it into solution. That's why there's no 0 point. So the idea is then you add some initial salt, and this increases the solubility, which they refer to as the salting in. And so in this case, for all of these salts, we see a decrease in the viscosity as we increase the concentration initially. But then at some point, the further addition of the salt is thought to decrease the solubility. And then this is referred to as the salting out. So ultimately, if you add enough salt, then you'll have something that crashes, the protein will crash out of solution. So we didn't get to that point on this plot here, but we're observing the change in solubility. I think we can correlate the change in viscosity to the change in solubility. And what we see is that the potassium chloride and the sodium chloride, which are very similar in the ranking of this series, behave in a very similar way. The larger deviations come from the extreme points on the series, that being the sodium bromide, which tends to be down on the level that they refer to as more prone to salting in. So in this case, the sodium bromide does give the lowest viscosity, and then upon further addition doesn't result in as extreme of an increase. And then on the opposite side, where we have the sodium sulfate, the sulfate then is leaning down on towards the end where they think it's more contributes more to the salting out. We can see that the increase in our viscosity as we add more and more of this then is more drastic than the other particular salts. And we did go to higher ionic strengths here, but the viscosity gets very high. So our highest viscosity we achieved here was over 1,000 centipoise. I didn't put it on there because it sort of diminished the ability to differentiate the other samples. But so this is the general view of what happens. And the thought is that the salt initially, from a qualitative perspective, the salt initially decreases the attraction, which promotes the salting in, and then further salt addition would increase the attraction, which then promotes the decrease in solubility and then the increase in viscosity. So also, what you find in the literature when you think about this process of adjusting the ionic strength is that viscosity in proteins tends to get very high in concentrated systems due to reversible cluster or aggregate formation, which is consistent with the idea of adjusting the attraction or the attractive potential. So one way to sort of look at whether or not this makes sense from a rheology perspective is to start looking at the shear rate dependence. Because when you start looking at shear rate dependence, you can start to probe characteristic time scales, which then correlate to a characteristic length scale, such as size of a cluster or size of an aggregate. So what we wanted to do once we got some of this preliminary data on the impact of salt as a function of concentration on viscosity is to take some of these specific samples and then look at the effective shear rate. And if we could learn anything about the sample from an analysis with this. So what we picked actually is the one that's most extreme, and this is the sodium sulfate. So I picked this is 75 millimolar ionic strength, and then I actually picked a point that's not presented on the curve because it's much higher. So it would be 1200 millimolar, which gives us a viscosity over 200 centipoise. So I'm picking two things, one down here where I'm starting to theoretically salt in the system, where I'm presumably reducing attractions, starting to reduce the likelihood of clusters, and then the other extreme case where I'm on the process of salting out or increasing attractions and then potentially increasing cluster formation. Okay, so we're going to pick those two samples and then look at shear rate dependence and see what we can pull out of there. So now what we're looking at are shear rate sweeps. So that's just the common way we refer to as viscosity as a function of shear rate. And we're all of the previous samples, as I stated, were for the 250 mg per ml gamma globulin. And so this is the same case, same samples. And we're just looking at two different sodium sulfate levels, the 75 and the 1200 millimolar. So initially, to start off with, I put them on a separate plot so that because the viscosities are significantly different, and I wanted to make apparent that we do have shear thinning behavior in each case. So there's different ways that we can start looking at this and extracting information from viscosity versus shear rate. One of these ways is just visually looking at the system. So if we look at the 75 millimolar and we look at visually where the onset of shear thinning starts to happen. So it's somewhere around maybe 300 inverse seconds or so, which is quite different than what we see for the viscosity, the onset of shear thinning for the higher salt content, which is going to be somewhere around 300 reciprocal seconds. So there's an order of magnitude different in the onset of shear thinning, what the critical shear rate is. And the reason that is relevant is because when you think of, and I think this will be a little more clear when we go into the next analysis part, shear rate is really varying shear rate is really probing different events on different time scales in the system. So a lot of times what you'll think of it as you're probing characteristic relaxation times of your sample. And so visually you can pull it off and you can see that, because shear rate is inverse time, if we have a onset of shear thinning occurring earlier, then that means the characteristic relaxation time is longer, which means that if we want to correlate to, say, diffusion of objects, which is, again, I think that'll become a little more clear in the next slide, then this would correlate to a larger object or perhaps cluster if that's a possibility in your system. So you can do it very just visually in that way. Again, remembering that shear rate is inverse time, and so this is going to probe these different time scales, and then that can correlate to a length scale. Or you could actually fit models and extract the model parameters and do the same thing in a slightly more quantitative way. So there are a couple of common models for viscosity as a function of shear rate. In this case, both of them work relatively well. One of these common ones is the cross model. This was developed more for dispersions, and then the Colloidal model, which was more for polymer solutions or melts. In this case, the dashed line in each plot is for the cross model, and the Colloidal is the solid line. And so what I have in the table are the parameters that we've extracted out for each model. And so if you look at the models, both of them have this coefficient lambda, which is going to have dimensions of time. And generally this is thought of as this characteristic relaxation time scale. And so if we look at either model, as we go from the 75 to the 1200 millimolar, what we're seeing is basically an order in magnitude increase in this characteristic time scale. Because the model form is different, the actual number changes, but the trend is equivalent, that we get an order of magnitude change, an order of magnitude increase as we increase the salt concentration. And so this is consistent with the idea of the salting out phenomenon, the increase in attractions, and then the more likelihood of forming clusters or aggregates. The increased relaxation time then corresponding to these larger objects. And then also, sometimes when you look at the cross model, it's been sort of interpreting the other coefficient, the extent, the rate at which shear thinning occurs, this parameter m. So it has an upper bound of 1 generally when doing the fitting. And the analysis of this is that when you have m equals to 1, that's closer to what is referred to as a monodispersed system. And as m goes down, the system is indicating to be more polydispersed. And so again, as the parameter m decreases, indicating increased polydispersity, this again is consistent with the idea of attractions and then aggregation, because when you have aggregations in this type of system, you don't generally form all doublets, all triplets, etc. You tend to form a spectrum of sizes, which then introduces effective polydispersity in the system. Okay, so another way to look at this is if we want to do some additional scaling and see if we can generate a master curve for this particular two groups of data. So again, we're looking at viscosity versus shear rate, and now we want to do some scaling to extract. Again, we're looking for a characteristic length scale that could represent a trend in the likelihood of aggregation. So what we're going to do is now we've taken the viscosity, and so that we have it easy to compare, we've scaled the viscosity on the low shear limiting value, on the plateau value at the low shear rates. And the top plot is still looking at this value as a function of shear rate. And so now again, it's very visible here on this particular plot that the earlier onset of shear thinning for the higher salt concentration, and then this is much delayed for the lower salt concentration. So what can we do here then to try to extract a parameter? So what we want to do is we want to scale shear rate. So if you think of, particularly in the colloidal systems, why do you have the onset of shear thinning? So if you remember from the very beginning when we tried to define the colloidal range, we made it clear that you have an upper limit on size because you still want to consider systems that have Brownian motion. So these molecules are going to be bouncing around in solution, and this is going to provide basically a resistance to the flow. And the shear thinning, the onset of shear thinning, can often be correlated to the point where the shear flow is going to overcome this Brownian motion. So initially, at low shear rates, the Brownian motion is sufficient to restore or maintain close to an equilibrium structure. But then at some point, the shear rate is going to overtake the Brownian motion, and then the material starts to shear thin. So how can we think of this in terms of timescales? So this Peclet number that I'm referring to is basically just a ratio of timescales, one of these being the characteristic time associated with the Brownian motion or diffusion. And so this our characteristic length scale that we're going to adjust here to try to shift these onto a master curve we're just going to define as L. And so this time scale is L squared divided by some effective diffusion coefficient. And so I'm just taking a diffusion coefficient that's based on the Stokes-Einstein law, but I'm modifying it for the fact that we have not necessarily an individual molecule, so I'm not using the radius like we normally would. And I'm also accounting for the fact that this is a concentrated system and diffusion will be slowed down due to the higher viscosity. So I'm introducing the low shear plateau viscosity, and I'm just sort of leaving this as a general length scale, not necessarily a radius. And then the other time scale relevant here is that associated with the flow field. And so this is just the inverse of shear rate. So we sort of already talked about that in a sort of a general way. And so the Peclet number is defined as this Brownian motion time divided by the characteristic time scale of the flow or convection. And so just to sort of repeat myself, when the Peclet number is very small, this means that the Brownian motion time scale, it's much faster. The Brownian motion is much faster than the flow field, and so it can respond, and it can resist the deformation and maintain the equilibrium state. But then as the Peclet number gets higher, then the shear flow starts to overtake this, and the shear thinning onset occurs. So to try to shift the data, what I wanted to do was adjust this length scale for our two particular samples that we're looking at here and see if we can develop a master curve and then what these length scales turn out to be. So in this case, I was adjusting the length scale such that the shear thinning onset occurred close to a Peclet number of 1. And so when I did this for our two systems with the different ionic strengths, I see that I had an increase in the length scale for the higher ionic strength system. So again, this is going to be consistent with the idea that as we increase the salt, that we're potentially introducing this salting out process and forming clusters or aggregates. And with this adjustment, I was able to basically develop a master curve with the two protein samples that I was looking at. Okay. So just then to summarize, I just wanted to introduce the idea with some examples of trying to relate viscosity to how the components are interacting and how the microstructure is coming about, and then relating this to the formulation components, just so you can sort of go full circle and understand, learn how to control and manipulate your rheology behavior. And I know that using this colloidal view is somewhat controversial for proteins. And admittedly, there are situations where it works better than others. But I think from a at least qualitative standpoint, this can support interpreting viscosity data and trying to understand it in terms of what's happening on the molecular level. So just a quick review then, it seems to be beneficial when we're looking at some of today's examples for, in this case, they are model proteins, the temperature stability, and then also the extent of aggregation that we get in the bovine gamma globulin as we introduce various salts and adjusting the salt concentration. So I will stop there and look and see if we have any questions. So I'll just wait a few minutes and see if things come in. Someone did ask if we do share the slides to everyone participating, and the answer is yes. We do send out a link in a few days of a recording of the webinar. So if you wanted to listen to the verbal part again, and then also the slides. So yes, you'll get the content, both the video and then the slides if you'd like. Okay, so it looks like we're not getting too many questions today, which I understand that was a little different presentation and material, and the analysis might be a little unfamiliar to people. So absolutely, we will send out all the information to you. And I think when you sign up, you get contact information for us. So if you, upon reviewing the information, if you have questions later on, please contact us. We'd be happy to discuss the information with you further. So at this point, I think I'll end. And again, thank you for joining us today.